Citrus Higgsbit webinar. It's a pleasure to have you here in this second session. To start this session, Dr. Stephanie Bloom, the Higgsbit coordinator and NAPO executive director, she will open this session together with the Agricultural Health and Food Safety Program Manager, Dr. Ana Marisa Cordero. Go ahead, Stephanie. And thank you very much, Lourdes. Good morning, good afternoon, everybody. My name is Stephanie Bloom from NAPO. I am the current coordinator of the HICSP, Inter-America Coordinating Group in Plant Health. I welcome you to this second citrus session. For me, it is a great pleasure to be here with you and have the opportunity to welcome our speaker, Dr. Beatriz Choconosle Cázares, who is a truly leader and a partner of this organization. I have a pleasure to work with Beatriz in the last 2 years, maybe more than that. Beatriz, you will remember how long we've been working together, but she is a very important person in one of our most significant projects, which is projects in NAPO, which is the diagnostic harmonization for seed-transmitted diseases. And we have the support from this wonderful researcher here in NAPO. And that's why I have a pleasure to welcome her, our coordinator today for HLB. He will introduce her. But I just wanted to share with those that are here that Beatriz is going to talk about citrus disease that are affecting our region, that have an impact in our region. I hope this session is wonderful, and I welcome everybody. Thank you very much, Ana Marisa. Hi, everybody. Good morning. Thank you very much, Stephanie. My regards to all those participants today. This is an effort, a group effort from the Inter-American Coordinator Group in Plant Health to under NAPO's coordination to make possible this series of webinar. We have a first session last week, and today we are seeing this effort. Thank you very much to Xavier, the coordinator of this group. Thank you very much as well to our speaker and expert today for her willingness and interest in participating in this initiative. Very good. This is part of the efforts that we're doing to try to share experiences, knowledge related to the inclusion of innovative technologies that will help us to respond in a best way to the current challenges. I welcome you, and I thank especially to you, and I wish you the best in this webinar. Thank you very much. Thank you very much, Anna. Thank you very much, Stephanie. Now, we are going to ask Dr. Xavier Uceda to introduce our speaker today. Xavier is a director honoris. He's the coordinator of this HICSP working group for HLB and is one of the main promoters of these activities. Thank you very much, Lourdes. Welcome, everybody. to this second session on citrus session. Before I start, I want to thank again to the Inter-American Coordinating Group in Plant Health, to the different RPPOs, because this has been a joint effort. NAPO, the Andean Community, COSABE, CAFSA, AICA as well, and OIRSA. Before I start, I would like to give some general announcements. First, with 2 sessions in this, you can receive a certificate and you can request it through the GICSV Secretariat. We can ask Lourdes, and for those who will require the certificate, can send an email to Lourdes, which is the one that we have on screen right now. I also would like to mention quickly that we have a good participation. It's going to be a wonderful closing of the sessions. We have participants from Dominican Republic, people from Peru, Paraguay, Fatima Alonso, some people with very important knowledge in Dr. Mari Carmen Sanchez in Mexico, who is familiarized with biological control. And I think we are covered at the continental level with excellent participation from well-known technicians. Now, I'm going to identify the objective of this second session for the citrus session. As you're aware, one of the main objectives in the first session is to share some techniques and processes related to decision taken for the citrus processing chain, early alert system, multi-criteria system, which we saw with Dr. Roberto Tapia, and some tools for dynamic alert system, as well as statistic for HLB, diaphorina citra. In this second session, we are going to, Dr. Beatriz, she's going to share the progress on biotechnological tools employed to mitigate the disease. As well, I'm going to read the summary of her biography, a summary of Dr. Beatriz's biography. As you are aware, she has a very extended CV, and we're going to just mention the highlighted version of this CV. I'm going to tell you that she's a biologist graduated from the National University of Mexico. She has a master's degree in genetics and molecular biology from CINSTEFAC in Mexico City. And she also has a PhD with major in biotechnology, as well from CINVESTAF in Guanajuato. She has carried out postdoctoral studies and took sabbatical leave in different countries, and is a member of the National System of Researchers at Level 3. She's currently an associate professor in the Department of Biotechnology and Bioengineering at CINVESTAN in Mexico City. And she has published more than 123 scientific articles, written several books and chapters. She has the 18 granted patents. She has directed about 50 graduate students and undergraduate thesis. She has over 5,800 times she has been cited, and she is very knowledgeable in technology, biotechnology in terms of citrus. I have the honor to welcome Dr. Beatriz, and we'll start with second session on citrus GICSV. I wish you success. Thank you very much. I'm going to share my screen.
Previo, doctora.
Doctor, before that, I also would like to mention that you can send your questions at the different social media platforms so that you're aware. You can send it through the Zoom chat as well as the YouTube channel chat, as well as the AICA channel. We have several platforms where we are and diffusing this, spreading this session. Thank you very much. Good morning, good afternoon. First, I would like to thank the Inter-American Coordinating Group in Plant Health for this opportunity to describe our research, and especially Dr. Stephanie Bloom and our NAPO colleagues, AICA or IRSA, for this organization. Uh, I'm going to move the different slides that I would like to share. Citriculture is an important economic activity in Mexico. We're the fifth country growing oranges, the first one growing limes. Consumption per capita is 15 pounds per year per lime per person and 80 pounds for oranges. Truly, Mexicans, we consume, we add oranges and limes to our foods. You can see on the screen street food and beverages based on citrus products, included margaritas. Very well. Years ago, we started a formal collaboration with Seneca, Mexico, to characterize emerging diseases. So Seneca regularly monitor our country. When field personnel identifies symptoms in plants or animals that current detection methods fail to identify the pathogen, together we take samples, we isolate the nucleic acids, DNA or RNA, do a massive sequencing, a bioinformatics analysis to identify the associated pathogens, and we develop a detection method. With its elements, Seneca controls or eradicate the outbreak. In this talk, I will describe our research on the process, citrus tristeza virus, and citrus screening. I am aware that Seneca monitor as well other quarantine pests. In 2014, Seneca collected samples in our country from Jalisco and Chiapas states, where the current methods were unable to identify the associated pathogen. However, the symptoms were consistent with citrus leprosis. After the massive sequencing and analysis, we identify a member of the Radiviridae family, and the viral genome is organized into RNA components. With this information, the phylogeny using the nucleocapsid or the RNA-dependent polymerase group the virus out of the nucleus or pseudohabdovirus clades and group them into the decorovirus branch. With this information, the virus was named as citrus necrotic spot virus or CNSV. The electron microscopy of the infected tissue could identify membrane, spheric viral arrangements, accumulating always within the cytoplasm, as well as chloroplast, with a likely viral protein forming a lattice. There were no virus observed within the nuclei of the 200 analyzed images. A little bit more about this virus. It is a non-systemic virus. However, it interacts with plasmodesmata, which are the intercellular communication channels to move from one cell to the other. However, it does not reach the plant vasculature. In terms of the massive RNA sequences analysis of the infected plants, it was demonstrated the induction of the secondary metabolites, maybe as a defense mechanisms against insect infestation, as well as the viral infection itself. Nowadays, Senacica monitors the vector and symptomatic plants. We all know this disease is present in Mexico, Central America, as well as South America. Please allow me to move now to the citrus stress virus. In 2018, orchard with disease orange trees were reported by Senacica, located in the highest production area in the country, which is the state of Veracruz and the south of Tamaulipas. Here we identify sweet orange trees infected with mild or severe CTB isolates, and they display different symptoms, including corky trunks and fruits with no commercial value. After a massive RNA sequencing analysis, we could identify 2 mild isolates and 1 severe one. When we compare the accumulation of the viral encoded open reading frames, so the protein of the virus, we found differential expressions between the mild and severe isolates. We were also be able to propose a diagnostic method based on the restriction with ECO-RI on the code protein amplicon of the severe isolate, which the save time instead of the conventional sequencing, which save time and resources. In terms of the differential gene expression, it allows us to identify induced genes in the mild as well as the severe. These genes have possible biotechnological application. We are already doing mining analysis data. And in this image, we can see the comparison with as asymptomatic trees that initially were selected as healthy material, but they weren't in fact, and we will see why. Please remember we're working with environmental trees, and we were able to identify readings of citrus exocortis viroid and the hope stone viroid. The asymptomatic trees, originally considered as healthy, in fact, had CEVD and HSVD. It's interesting to highlight that the severe strain, which is always present in both viroids, we found it in this combination. On the other hand, the mild strain doesn't have these viroids. We still need to analyze more trees to confirm these findings, since we only analyze 20 of each condition. So we're working on this so far. Now I move to the piece of resistance, the citrus screening. Our very favorite research. We're very passionate about this. Citrus screening is known in Mexico as HLB and is currently present in all our orchards in Mexico. The bacteria associated with HLB is the Candidatus labibacter asiaticus. It cannot be cultivated, and it resides in citrus vasculature. When this happens, it blocks, as you can see here, the infected plants, the normal movement of photoassimilates, and to control the bacteria that infect the plants, they synthesize antimicrobial proteins, which are detected in the exudates. The microbial proteins that are in the exudates are not present in the proteome of the phloem sap, where the bacteria reside.
A través de fusionarlas con una proteína acarreadora en la savia del floema. En teoría, la proteína móvil CSPP16, unida covalentemente al antimicrobiano, facilitaría su transporte a la vasculatura, donde crece el patógeno Candidatus liberibacter. Así que la hipótesis fue que la presencia del antimicrobiano dentro del floema.
Beatriz,
Sí.
Discúlpeme por favor la interrupción. Tenemos un pequeño problema que en el canal de español se está escuchando en inglés. Nos da un segundo, por favor, y nos disculpa la interrupción.
Claro que sí, muchas gracias.
Y mientras recuperamos la comunicación ya en el canal que corresponde, doctora, mencionarle que en la contabilización que tenemos de
mention that according to our books and the countries that are joining us, we have 18, 19 countries according to our count. So this is a success, the second session, and this is for your technical backup that we are receiving. So congrats, kudos to you. The reception is great. So Lourdes, could you confirm that we can continue? So sorry. We apologize for the interruption. Oh, thank you so much for the excellent organization. Let me then pick up from here. So what we proposed is that the presence of AMPs within the phloem would decrease the bacterial content because AMPs poke holes in the bacterial membrane and therefore they cause bacterial death. We have worked with systemic proteins and RNA for 25 years, and we know these proteins are transported to long-distance tissues using the vascular system, and they can move from stalks to scion plants. CMPP16 is a supracellular protein which is present in the phloem sap. And here you can see some images where it is identified in the vascular tissue. Now, supracellular proteins reach distant tissues, as we said, to fulfill their function. For example, adaptation to abiotic stress, phosphate allocation, tuberization, insect attacks, and so on. In collaboration with the group led by Dr. Lucas in California, we demonstrated that flowering is induced by systemic transport of FT protein in pumpkins, beans, and Arabidopsis. And here you can see a tiny plantlet mobilizing FT, which is already flowering. Now, with this background information, we designed mobile AMPs to reach the citrus vascular system. In citrus, we identified the same proteins we had characterized in other plants before. And here you have PP16 in citrus. It's present in the phloem sap. It is identical in oranges, limes, and lemons. And we identified other similar or equivalent proteins in citrus phloem, as you can see on this slide. Now, the interaction of these citrus proteins is similar to those observed in pumpkin, as was determined in bioinformatic analysis. And here I have the little truck interacting with its counterparts in citrus. We started employing the human defensing to prevent immune responses in consumers, but we also used other natural AMPs and newly designed proteins. I'm showing you a cultured Clavibacter michiganensis bacterium, which was controlled using our antimicrobial proteins. These are the genetic constructs employed. On the left, you can see B defensing, lysozyme, meganing, sacropin, and fusions we concocted with the fluorescent protein GFP to trace its movement. All of them were driven by the strong promoter P35S. On the right, you see similar constructs, but they were driven by the vascular promoter P5880. These are the citrus transformation methods we have in our lab: bioballistics, agrobacterium, and recently, pollen magnetofection. In this slide, you see the detection of transcripts and from their fused proteins in GM citrus, and you can observe these signals in the vascular area. CSPP16 and AMP fusion accumulates in companion cells and sieve elements, which are two specialized cells in the plant vasculature. We can see here a beautiful vein full of green fluorescent signals. Once we got these plants, we wrote our dossiers containing all the technical information. We requested a federal permit for an open field trial in Colima State on the Pacific. This state is the top producer of limes, and it's been hard hit by citrus greening. Here you can see the area, and you can tell the trees which are growing and the controls that have been carried out. Now, AMPs mitigate citrus greening. We are aware there is low bacterial content in late spring when temperature reaches 45 degrees Celsius, which translate into 113 degrees Fahrenheit. There are other AMPs that are also useful, but public opinion frowns upon their use. There are also other alternatives to GM. Sorry, you're here looking at some aspects of the open field trial. These trees are productive, although perhaps you can see in old leaves a certain diffuse mottle, which is associated to HLB. However, fruits are of top commercial quality. Let me show you here the expression of AMPs in oranges and Persian limes with good results as well. And to address the comment of an environmentalist on the impact of non-target beneficial bacteria, we conducted metagenome studies on soil, and we concluded that bacterial diversity was not affected by GM citrus. The observed variations are due to the season, not to the treatment. Very well. As an alternative to GM trees, we obtained AMPs in chitosan nanoparticles, as you can see, and we demonstrated their capacity to inhibit the growth of Clavibacter michiganensis, but we did need a special permit to analyze nanoparticles in open field trials. And we are filing our requests. As an alternative to GM as well, and to use this editing tool, we are editing the citrus genome to translocate citrus defensing to the CSPP16 locus. This would result in a similar product, and it would endow defensins with mobility so that they can penetrate citrus vascular tissue. We are analyzing microbiota of insect vectors in order to handle the content of microbiota, to be able to work with it. In closing, I would like to say that monitoring emerging diseases can stop and pause their dissemination, but they can also stop their evolution to more virulent forms. And this translates into many examples, which includes COVID. It is possible to endow proteins and RNA with supracellular properties. That's part of what we do in our group. And supracellular AMPs translocate into the plant vasculature can indeed mitigate HLB. I would like to thank you once again for this opportunity. Let me show you the members of the lab who worked on the results I have presented to you today. This group is also coordinated by Dr. Roberto Ruiz Medrano. And on your right, you can see our contact data. And here I am, ready and willing to address your valuable comments and suggestions. And I thank you for your precious attention. Thank you so very much, Dr. Beatriz. We invite participants in this second session to send us their queries, their questions, send them over the chat. Now, some of the questions that we have received so far. Dr. Sarmiento from Panama mentions that, well, he sent me a whole text, but I'll try to summarize his comment. AMPs, when they were trying to manage HLB, didn't seem to be a feasible solution or an efficient one. So he's wondering whether you could elaborate on the actual status of AMPs with regards to the expression of symptoms or the production of an orchard infected with HLB. Yes, thank you. In fact, other groups looked for a way of employing AMPs. It was a straightforward idea, it seemed so, but because of their nature, their biochemical nature, AMPs could not move into the phloem. So our research sought to provide that systemic movement so that the molecules could penetrate where the bacteria was residing so that they could make a difference for the better. I do indeed consider that if we can manipulate the movement property of a molecule, we are on the right path. Before I give the floor to Mr. Jose Hernandez, let me ask, is there any residual factor in the fruit that could be detected in some echelon of the productive chain? Indeed, there is. If we have a GM plant, it is possible to identify both the codifying gene, its transcript or messenger, and the protein. However, if we use GM stocks and science without genetic modification, we cannot detect anything. This is an alternative we have considered, thinking of exports, of course. Thank you. And we share the floor with Dr. Hernandez from NAPO. Jose? Thank you so much for your excellent presentation. It was very interesting. And my question is, in what stage of this research related to a trade use are we? Is this feasible to apply to great cropping areas, great producing areas? Because it would take a lot of work to produce AMPs. So I'm wondering about the practical aspect. Is it feasible at all? We do have an experimental area. Technically, we can obtain a trade permit after complying with the Biosecurity Act, which is quite complex in Mexico. If we could move on and have commercial lots, then we would suggest reconverting orchards through using materials that can mitigate or resist HLB. These are great samples. We have great examples of this. Would this be a kind of cure? Would this eliminate the infection? Could we recover the plant so that it could yield a better crop? In fact, in our country, reconverting orchards takes place quite often, and we could include these materials in any natural reconversion system used currently. We're talking about 16, 18 years, and that's when we could introduce the new materials. Excellent. Thank you so much. Some questions we have received through our YouTube channel. Ronald Leonardo Chique Morales asks, this treatment would eliminate in the vector the capacity to infect other plants? Thank you so much for the question. The answer is yes. And we've been interested in evaluating the vector because where we have experimental plants, we found insects with a low content of Candidatus liberibacters, and we wondered why. So we're thinking of taking our microbiota, that is the subject of our studies, and couple it with beneficial microorganisms, with plants which are producing naturally these AMPs. But this would be a major element to control the vector, even more perhaps than using pesticides that we have to this date.
We have another consultation, Dr. Alonso Sasso, and he says, HLB, what is the stability of the microbiome once it's produced in the plant? And how feasible can the GM be manipulated so that the vector is less effective? Beatriz, yes, it's a very interesting question. Thank you for this question, Alonso. The plant is synthesizing the AMP. So the accumulation, let's say, will depend on the metabolic rate of the citrus. So if we think in the number of hours of heat that we have in the experimental area where it's very tropical, we think the accumulation is very important. However, from the analytical point of view, we had needed semi-purify or concentrate the extracts in order to have a Western blot detection. The protein will be there. And thinking about the vector, we still need do more experiments. And this is something we are integrating, trying to get into, to see it in a more holistic way, the bacteria, the plant, and its vector. Xavier, thank you, Doctor. And now that you're speaking, I have my own question. You mentioned the trial in Colima. And in terms of the use of the AMP, do you have any difference in oranges crop as opposed to lime crops in how the AMP behave? Yes, we found in lime, we have different varieties, some more susceptible than others, but definitely oranges is more susceptible, are more susceptible. So we can also consider that is due to the high homozygosis of the crop. And this is another alternative the biologists are proposing, to be using different genetics background in order to, from the genetic point of view, to be able to have more variability in alleles in order to protect the crop. Not only this one, but other crops. So going back to oranges, yes, it is more susceptible. Very interesting, says Xavier. Mr. Jorge Carlos Ibra from in the YouTube channel mentioned that if there have been any trial for citrus to control citrus stress virus. Beatriz? Really? No. We would like to, however, to continue exploring the presence of viroids. Because when we identify several plants from the technical point of view, which have healthy plants that have the viroids, it get our attention. There are examples of other virus latent infections which have no symptoms, show no symptoms in plants. And if we could have a vaccine, in quote unquote, to protect the crops, it would be a very interesting question for us. That's what we have thought in our group, but we need to further discuss with our counterparts to see how we can implement it and to have a closer idea on the possible implementation and the possibility to have a resistant genotype. Xavier, thank you, Dr. Sokonostle. I think that's all the questions that we have in the different platform. I would like to mention that we have representatives from Venezuela, Honduras, from Nicaragua, Panama, other colleagues from Nicaragua, Belize, El Salvador, Rosa Maria Martinez, Argentina, Paraguay. We are not going to mention all the list, but what I want to mention is that we have so far technicians or experts representing over 20 countries from the American continent. Dr. Beatriz, you have any final comment, any recommendation? Now that you participate in this second session, to conclude with this citrus session. I also want to thank you, and I would like to share that biotechnology is something that moves the economy of the developing countries. I think it is a tool that we should apply with more emphasis in our region because our borders are very artificial. We are sharing these problems, and together we can, with the new tools, we can try to get closer to their solution. Thank you again for this wonderful opportunity. I reiterate that I'm at your disposal. Thank you very much. Thank you. We are inviting you to later on. You can participate in a second session because the topic can go further, and it have awakened interest from the participants. We invite you again to a second session. On behalf of GICSV and on my behalf, I want to thank you for your participation. And we have the final message from this second session. To the closing word for Lourdes or Stephanie, Dr. Beatriz, thank you very much for your kind participation. Beatriz, thank you very much. It was my honor. Thank you, Javier. Lourdes, I want to thank from the bottom of my heart the excellent presentation, Beatriz. Whoa, even myself as an entomologist, I understood it. So I thank you to do the molecular biology accessible to people like me. The solutions are innovative, and I wish you the best in your research. It is very impressive, your work. Thank you very much. Lourdes, go ahead. Thank you, Stephanie. From ICA, I want to thank Dr. Beatriz for your effort, and as Stephanie mentioned, she explained whoever knows their topic, they explain it in a simple way. Thank you very much. For a second, I want to share the email where you can request your certificate of participation. I would like to thank all the participants that were on the social media platforms, as well as Zoom. This is another GICSV effort to coordinate phytosanitary strategies. I would like also to thank a special thanks to all those who make this event possible. I would like to thank especially Viviana Chaco, Mrs. Picado, Adrián Altizar, that behind the scenes, they were made possible this transmission. Thank you very much. We will see you very soon. Bye for now. Greetings, everybody. Bye.